Respected teachers and learned audience, Assalamu alaikum and a very good morning to you all. I am Dr. Abir Hassan, Phase B resident, Department of Dermatology, BSMMU, is going to present today's presentation. Our topic of presentation today is Itraconazole in dermatophytosis, tackling recurrent and resistant fungal infections. Dermatophytosis or superficial fungal infections are one of the most common conditions encountered in our outpatient department. Emergence of resistance and recalcitrant fungal strains have made the treatment of dermatophytosis very challenging for the dermatologists. So, the judicious and proper use of antifungals are therefore the cornerstone of successful treatment. We can broadly classify the fungal infections into 4 subtypes, namely the superficial fungal infections like pityriasis versicolor, the cutaneous fungal infections like dermatophytosis, subcutaneous fungal infections like chromoblastomycosis and sporotrichosis, as well as the deep fungal infections like histoplasmosis and blastomycosis. Our today's highlight will be basically on dermatophytosis. Dermatophytosis is a cutaneous fungal infection which mainly affects the skin, hair and the nail, and it is caused by the following fungus: Trichophyton, Epidermophyton and Microspora species. Recently, Trichophyton indotyne has spread out widely among the Indian subcontinent and has become a great cause of concern. The dermatophytes can affect different parts of our body and we can name them accordingly. For example, if it affects our head, we can call it tinea capitis. Similarly, tinea facie in the face, tinea corporis in the body, onychomycosis in the nail, tinea pedis in the feet, tinea cruris in groin, as well as tinea manum in the hands. Now, I would like to draw your attention to a more specific and the recent outbreak that has caused a lot of havoc nowadays. We call it Trichophyton mentagophytes type 8, or in other terms, we know it Trichophyton indotyne. It was first reported in 2008 in Australia. In 2020, it was identified as a separate species. It's a zoophilic genetic origin with a person-to-person transmission, and the most shocking and the most important thing is it is in vitro terbinafine resistant. Now, a few words regarding its drug resistance. It has been shown to be resistant to terbinafine in vitro in about 20 to 70% cases, mainly due to the genetic mutation of squalene epoxidase. Whereas in case of itraconazole, it has been shown to be resistant in vitro in up to 25% cases. The more dangerous thing is that there are new strains with double mutations. That means it is resistant to both terbinafine as well as azoles. Now, regarding the treatment options of dermatophytes, we can broadly classify it into 2 broad headings: the topical as well as the systemic preparations. In case of topical preparations, there are a lot of options that are available for us. However, the azoles are the preferred first-line topical agent for dermatophytosis. Among them, in the recent guidelines, it has been shown that luliconazole is the preferred drug of choice in India. Some few local studies have shown that there are low MIC values for both miconazole as well as clotrimazole. Cyclopirox as well as amorolfine has also been shown to be effective in dermatophytosis. However, there are lack of studies regarding combination of 2 topical medications, so we should not prescribe it until there is a strong recommendation about it. Now, when we should use topical medications? In case of naive tinea or the first onset of tinea, which is single or with a very few lesions, if the lesions are small, in case of first trimester of pregnancy, where we have to be very cautious about systemic medications, or if there is any contraindication for any oral medication, topical medication can be our first line drug. It should be mentioned that the topical steroids in any forms, either mixture or components, are strongly discouraged in dermatophytosis infection. There are some antifungal soaps available in the market. However, the recent guidelines say that the antifungal soaps are not recommended. Now, in case of the oral treatment preparations, there are a few options available like Itraconazole 100 mg BD, terbinafine 250 mg daily, fluconazole 150 mg daily, and griseofulvin 500 mg daily. Among them, our focus will be on itraconazole today. Now, regarding the duration of treatment in dermatophytosis, there are two opinions. In the standard textbooks, it has been written that for tinea capitis, we can use it up to 8 weeks. For tinea corporis, up to 4 weeks. In case of onychomycosis, we can use it up to 12 weeks. However, the recent Indian and the Sri Lankan guidelines for the treatment of dermatophytosis differ in this manner. They have recommended to use it until the cure, and in some cases they have recommended to use it at least 1 month after complete clinical cure. Now, in case of trichophyton indotini, there are a few treatment options available. Itraconazole 200 mg daily is the preferred choice. Although terbinafine has been shown to be resistant, but we can use it in longer duration and we can combine with other topical medications for benefit. Fluconazole and griseofulvin has been found to be ineffective in trichophyton indotini. Now, this is an article which was recently published on trichophyton indotini and regarding its drug resistance and the treatment options. And the options that I have already shown are already mentioned in this article. Now, we all should know about recalcitrant dermatophytosis. In this heading, there are actually 3 subheadings which we all should know about. One is the chronic dermatophytosis, which means persistence of tinea or dermatophytosis for more than 6 months, either continuous, recurrent, with or without treatment. Another type is recurrent dermatophytosis, which means that reappearance of the lesions 4 weeks after stopping the treatment following clinical cure, whereas resistance means persistence of tinea with poor or no response to standard dose and duration of the treatment. In all 3 cases, itraconazole is the preferred treatment of choice as per the national guideline of India and Sri Lanka. Now, we can broadly classify the antifungal drugs into 4 main headings: polyenes, azoles, allylamines, and echinocandins. And our focus of today will be on the triazole group, mainly itraconazole. As we can see, this is the structure of a fungal cell, and we can see that the fungal cell membranes are mainly consists of ergosterol, and which is synthesized from squalene. The azole groups mainly inhibit the synthesis of this ergosterol from squalene, and thus they break down the fungal cell membrane, and thus they insert their antifungal actions. Now, regarding itraconazole, it is the first generation triazole which was synthesized in 1980. It has a broader spectrum of action compared to other azoles. It is fungistatic, but it is equally effective in immunocompromised and immunocompetent people. It has a bioavailability of about 50 to 60%, which is widely variable from person to person. It varies depending upon the fed as well as the fasting state, and it is more bioavailable when after food intake and after increased gastric acidity. So, it should be taken after food. However, there is a preparation called oral suspension which contains cyclodextrin components which are better absorbed in empty stomach. It has a plasma protein binding of 99%, half-life of 21 hours. It is metabolized in the liver by cytochrome 3A4 enzyme, and it is excreted about 40% by the kidney and the rest by the feces. The most important thing to note is that it has a non-linear kinetics. That means that after administration of each drug, a fixed portion rather than a fixed percentage of the drug is excreted. As a result, there is increased chance of toxicity if we increase the dose. Now the mechanism of action, it mainly inhibits the 14-alpha D methylase enzyme, which inhibits the conversion of lanosterol into ergosterol, and which is an essential part of our fungal cell membrane. Now itraconazole is available in the market in these 4 formulations: the capsule 100 mg, tablet 200 mg, oral suspension 50 mg and 5 ml, as well as the newly launched super bioavailable capsules 65 mg. These are the indications of itraconazole. As we can see that dermatophytosis is currently an off-label indication for itraconazole. Some adverse effects which we all should be aware of while prescribing itraconazole. It has an unpleasant taste, mainly the tablet formulation as well as the oral suspension causes it. Anorexia, nausea, vomiting. In case of elderly, it can cause a triad of edema, hyperkalemia and hypertension. The most important thing here is a negative inotropic effect. So it has an effect on our cardiac cycle. So we should be more cautious while prescribing it in patients with heart failure, especially in elderly people. It can also cause us peripheral neuropathy as well as adrenal suppression. It is pregnancy category C, so you should be aware of it while prescribing it in pregnancy. It also excretes through the breast milk, so it should be avoided during lactation. Now the use of itraconazole in case of recalcitrant dermatophytosis. There are a few articles as well as some few case reports available regarding the use of itraconazole in case of recalcitrant dermatophytes. This is a journal which was recently published, which was showing the selection of itraconazole in the treatment of recalcitrant dermatophytosis, especially the trichophyton indotini. And this article showed that itraconazole was clearly superior to both terbinafine and fluconazole in the treatment of recalcitrant trichophyton indotini infections. This was another article which was recently published, and this highlighted the dissolution and the efficacy of itraconazole and its relevance to recalcitrant dermatophytosis. And it showed that the formulation of itraconazole is also very important in achieving the success. It showed that a small bead size, optimum drug polymer ratio, more number of pellets, as well as film forming polymers and thinner coating results in a faster dissolution rate and higher bioavailability. And all these points we get in super bioavailable itraconazole. This is another article which was recently published regarding the use of super bioavailable itraconazole in the management of recalcitrant dermatophytosis. It was the consensus statement by the dermatologists, and they have given us a guideline of when and how much should we use super bioavailable itraconazole in dermatophytosis. This is a simplified chart which shows that super bioavailable itraconazole 130 mg single dose for about 6 to 8 weeks was shown to be effective in the treatment of recalcitrant dermatophytosis, and they have recommended it to use it at least 2 weeks after achieving complete cure. This is another journal which was recently published by the Indian Academy of Dermatology and Venerology regarding the use of super bioavailable itraconazole in case of recalcitrant dermatophytosis. And they have given us a 15-point guideline regarding the use of super bioavailable itraconazole. The summary of this presentation was: resistance to itraconazole is not widespread as of now in India. The conventional itraconazole preparation shows considerable intersubject variability in pharmacokinetic parameters. The bioavailability of conventional itraconazole considerably varies when administered under fasting and fed conditions. Super bioavailable itraconazole utilizes the novel drug delivery technology for controlled release of the drugs in the upper duodenum, bypassing the requirement of acidic gastric pH, which makes the absorption of super bioavailable itraconazole less dependent on stomach environment. Super bioavailable itraconazole also demonstrates considerably lower intersubject variability in case of pharmacokinetics compared to the conventional itraconazoles. So, the dermatologist should advise the administration of super bioavailable itraconazole under fed condition as like the conventional itraconazole. The population pharmacokinetics study has predicted the 58 milligram of super bioavailable itraconazole was equivalent to 100 milligram of conventional itraconazole. As itraconazole follows a non-linear kinetics, the repeated dose of pharmacokinetic studies from the manufacturers should be taken into consideration. So, instead of increasing the dose in a single dose, we can divide it into 2 doses for better pharmacokinetic properties. Co-administration of itraconazole with proton pump inhibitor may affect its absorption and can reduce the overall bioavailability, whereas co-administration of super bioavailable itraconazole with PPI can increase its overall exposure. So, if we have to prescribe PPI and there is no alternative, we can use super bioavailable itraconazole along with the PPI. This is an advantage. The adverse event profile of super bioavailable itraconazole has not been studied separately by the manufacturers. So, the dermatologists had to screen the patients for all possible adverse effects similar to conventional itraconazole. As of now, there is no robust clinical trial available to establish the superiority of super bioavailable itraconazole over conventional itraconazole in terms of better cure rate or lower relapse rate in the management of dermatophytosis. However, the better pharmacokinetic parameters of super bioavailable itraconazole need not to be translated into a better cure rate or lesser relapse rate of dermatophytosis, and hence the dermatologists must build upon their own experience in this regard till more data are available. So, the take home message for today's presentation is itraconazole is the drug of choice for recalcitrant dermatophytosis. It is also the drug of choice in dermatophytosis by Trichophyton indotini. Super bioavailable formulation provides consistent pharmacokinetic property than the conventional itraconazole. The ideal duration of treatment should be 2 weeks after clinical cure, and it should be used cautiously in elderly patients due to the risk of hypertension, edema and hyperkalemia.
I must be very thankful to the previous speaker, Dr. Abir, because he has very successfully given out the physiology, pharmacology, bioavailability and other aspects. And he also touched some of the pharmaceutical technological aspects. Now, as a pharmaceutical technologist, as a pharmacist and pharmaceutical formulation experts, it is our duty to give the indications of how these types of technology works and what advantages they might pose over other dosage forms. So, my topic today is super bioavailable itraconazole, a paradigm shift in drug delivery system. As we can see, itraconazole is the triazole antifungal medications used for the treatment of diverse fungal infections. You can see the structure. Actually, in pharmacy, all the drugs are classified into 4 groups based on the solubility profile and the permeability profile. We must know that the solubility profile can be taken care by the formulators, because we can change the solubility of a low soluble drug by different techniques, by using solid dispersion, by incorporating the molecules into beta inclusion complexes, by making co-precipitation, by incurring some groups which will make the molecule, render the molecule more soluble, or so many other techniques are there. So, first of all, we have to understand that this molecule in particular, itraconazole, belongs to BCS class 2, biopharmaceutical classification 2. What does it mean? It actually means that it has got a low solubility and high permeability. We are very happy about this because permeability is a God-gifted thing. Permeability is through the gastrointestinal membrane. There are so many mechanisms, transcellular, paracellular, endocytosis. There are different mechanisms. There are some revolving door theory and other theories. There is involvement of super enzymes, cytochrome P450, etc. And it crosses the membrane and goes to the systemic circulation. And we all know that some of the drugs, maximum of the drugs sometimes go to the portal, through the portal vein goes to the liver, and sometimes get metabolized over there. Sometimes we get some benefit that some prodrugs can be active in the liver, but in the most of the cases the drugs are being excreted, that is not useful. So we have to take into consideration before we are formulating a drug. There are some common things are there. We know that 70% of the drug of this world is weakly basic, 25% drug is weakly acidic, and 5% drug is zwitterion. This is the first proposition. Second proposition is that acidic drug ionizes in basic pH, and basic drug ionizes in acidic pH. And third proposition is that the ionized drugs are water soluble. The ionized drugs are water soluble, and the non-ionized or unionized drugs are lipid soluble or organic solvent soluble. So our concern in case of oral is the gastrointestinal tract, and we will focus on that how we can exploit this body system to get the drug in our way, because there are USP means United States Pharmacopoeia, British Pharmacopoeia, and other compendial requirements for the drug release pattern in vitro and in vivo. So I will talk more or less on all these things. Another thing is that these days we are concerned about the rule of 5. That is called Lipinski rule of 5. Whenever one molecule is come to us, when we went for our PhD or other degrees, the supervisor gives us the molecule, and then we have to understand that the molecular configuration of the molecule means this that I told that the rule of 5 means every molecule has to satisfy certain properties like its solubility and permeability profile, which is partition coefficient, its molecular weight, the rotatable hydrogen bonds on it, and so many parameters we play with. Okay. The itraconazole itself is a highly lipophilic molecule, and it is slightly acidic salt. pK is 3.7. Means we can understand that it is more or less towards the basic drugs. Let me give you one simple example. Diclofenac sodium, acidic drug, and it does not dissolve at all. Only 10% diclofenac sodium may be dissolved in the stomach because the pH is acidic in nature. And I already told you, acid does not ionize acidic drugs, so it remains mostly unionized. On the contrary, say for diltiazem hydrochloride, it's a basic drug and it shoots in the stomach because most of the drug gets ionized. And I told you the ionized drugs are water soluble, the unionized drugs are lipid soluble, means we ultimately need the drug to be in its unionized form to cross the lipoprotein barrier. Now, itraconazole is highly lipophilic in nature, slightly acidic, etc., and hence is ionizable only at very low pH values. It is also poorly soluble in water. As I told you, it is biopharmaceutical classification 2 with an aqueous solubility close to 1 nanogram per milliliter at pH 7 and slightly higher than 4 nanogram per milliliter at pH 1. From these statements, it is clearly evident that itraconazole will ionize and be solubilized better in lower pH. These characteristics make the oral absorption of itraconazole difficult when it is administered as capsules. For this type of formulation, reduced bioavailability was observed when the drug was administered under fasting conditions. Now, the mechanism have been covered very nicely by Dr. Abir, and his presentation was excellent. And as you can see that itraconazole is interfering with the lanosterol 14 alpha demethylase. Itraconazole interferes with the cytochrome P450 dependent enzyme, and this results in a depletion of ergosterol in fungal cells, which ultimately leads to disruptions in membrane structure and function. So this is the basic mechanism that itraconazole interferes to the formation of ergosterol, which is deemed necessary as an antifungal agent. I will talk now some of the pharmacokinetics behavior, and then I will go to the most expected that you expect from me, the formulation aspects that will be my last part. So a little information on the pharmacokinetics, that is, we know that as we know that there are two terms. One is pharmacokinetics and another term is pharmacodynamics. Pharmacokinetics means what the body does, what the body exposes, what the body invites the drug is the property of the body, means ADME, absorption, distribution, metabolism and excretion. These are our physiological profiles, differs from country to country, man to man. And that's why you know already genetic polymorphism and specific drugs are now being given for the specific treatment. So, and pharmacodynamics is the sole authority of the drug. It's the pharmacological dynamics of the property. The drug should act on the cellular level, right? So that is the pharmacodynamics. Now, some of the pharmacokinetics property we need to know. The gastrointestinal tract absorption of itraconazole is about 55%. Peak serum concentration is 0.1 to 0.2 milligram per liter after 2 to 4 hours later. Itraconazole has high protein binding capacity, and then itraconazole is extensively metabolized in liver by CYP3A4. And the half-life of itraconazole is from 16 to 28 hours after a single dose. We will not go too much details of this, but here actually I can see that one brand which is called Tolsura, they are actually experimenting with this SUBA technology, super bioavailable technology, and the conventional dose is 100 milligram, whereas with only 65 milligram we can meet up all those things. And there are some parameters. Actually, after the drug gets into the blood, it is called bioavailable. We must know that not all the drugs are bioavailable in the body, but fraction of drug is bioavailable, because I already told you some of the drugs will get lost through the portal vein and some other parameters, and there are some phenomenon. Some drugs will be lost in the stomach because of solubility and ionic and non-ionic parameters. I will give you details of that. So there are some parameters on the area under curve, because ultimately there are two terms is remaining. One is called bioavailability. Bioavailability is a complete package. When a new drug is coming to the market after expenditure of $1 billion by the big pharma companies, they used to take a molecule, they used to take consideration what are the problems, what are the challenges. Is the molecule already resistant? Do they need to molecularly modify with some groups that molecules, or they will go for a new drug from a new plant source, mineral source, etc. So new drug they will take, and then they will go for the pre-formulation studies. Their solubility, their microbiology, their pharmacology will be tested, and long-term test with the animal will be started. That is called preclinical. At that stage, they will go for IND, investigational new drug application, to the US FDA, and then FDA will come and then check up and progress, and they will suggest for the clinical trial, which is phase 1, phase 2, phase 3, you all know. And after all this completion from biological and other viewpoints, the pharmaceutical scientists also go for technological advance, that is the source and procurement of the raw material, and then scale up technique, the environmental management system, good laboratory practice, current GMP, CGMP, All these things will be incorporated, and US FDA, when they come for a particular drug, then they, in Bangladesh also, they go for strict and stringent activity so that they can get the highest out of it. I'll talk later. So these are some absorption profiles that have been covered. I'm not going too much for this. This is a graph. We can see that the blue color one is super bioavailable itraconazole, and the yellow one is conventional itraconazole. And as I told you that this graph will not signify everything, but what I understand is that the conventional dose is 100 milligram or twice daily if it is then 200 milligram, and sometimes as tablet doses from 200 milligram. But this new technology will give everything within only 65 milligram. The area under curve, the superimposing of the curves. One thing I must share. So when a new drug is coming, the US FDA is going for all the parameters, including its dissolution in vitro, dissolution in vivo, its MDT values, mean dissolution time, and then going for safety parameters. Even they go for the post-marketing surveillance, the adverse drug reaction, etc. Everything is being monitored in a new drug. Now, after 20 years, the patent expires. Patent means the exclusivity, means the brand. As for example, the brand of brand is only given with the US FDA for new drugs. After 20 years, from 15 years, the normal pharma companies go for reverse engineering. They unfold the technology of the parent company, unfold the technology, how the granulation was done, how the tablet was made, what was the mixing order, what is the source of the raw materials. Everything in vain is taken care by the scientists, and after reverse engineering, they do understand the technology adapted by the parent company, and then they go for with this technology, prepare the same dosage form, and they then only need to give the AUC or bioequivalence. They don't need to go for preclinical, clinical, other safety parameters, because that is already been established by the parent company. So these companies called generic companies, they will be able to sell their drug with the name Itraconazole, not with any other names. But in Bangladesh, the scenario is different. We normally here, we use the local marketing brands. Limitations of conventional itraconazole: less bioavailability, 55%, less absorption, less efficacy, patient to patient dose variation. Yes, we'll take care, and we'll take care of these points, because on these points our formulation strategy will build on. Proton pump inhibitor use hampers the absorption, means co-administration of any proton pump inhibitors. Unabsorbed drug accumulation in the intestine, and then less bioavailable when taken in empty stomach. So this is, now I'll talk about the SUBA technology, which have been approved by the US FDA, Australian TGA, and other European Medicines Agency, UK MHRA, etc. Science of this technology. The novel SUBA process utilizes a unique spray drying technology. What is a spray drying technology? In a spray drying machine, we actually administered with force through a nozzle the solution of our itraconazole with the polymer we want to form the matrix. So the drug and the polymers will be mixed, and then it will be forced through a nozzle. It will come as a spray, and then we will have the source of dry air, and in a cone there is a cyclonic movement. In a cone, these particles, these spray dried particles. What is the spray dried particles? At the very beginning, it is water droplet, because inside the water there is the polymer and there is the drug. They are mixed as a physical mixture. These spray droplets will then go to the cyclonic zone, and along with the help of the dry air that will be dried, and we'll get the spray dried particles that may be few nanometers size, etc. And I have seen some of the technological things were shown in the previous slides: pelletization, microencapsulation, microspheres, nanoparticles. So many things are there for particular drug delivery and drug targeting purposes. That's another chapter. We'll talk it later on. So we'll go for the novel super process utilizes a unique spray drying technology which combines the drug and the polymer solution to generate an amorphous solid dispersion powder. Please, we have to concentrate the term amorphous. Mostly in conventional processes, itraconazole is given as a crystal form. A crystal is an organized soldiers. Why? Because as per the Bragg's equation, it is n lambda equal to 2d sine theta, as you know. It is the crystals are so organized. It has got interplanar spacings, and they are very hard to break. That's why their solubility is very, very low. That's why we cannot desire too much expectation from a crystalline dosage form. That's why when this polymer is mixed and then it is spray dried, the total structure changes into amorphous form, means the molecules are now disorganized, and their water solubility increases manifold. So this is the main technology, and this is the technology to increase the solubility of itraconazole. As I told you, it is a BCS class 2 drug. Now we have to have other considerations in our mind that we will go for. The technology and spray dryer, as you can see, dry air and the drug and polymer solution, I have explained it already, and that will come as a fine spray because you are forcibly pushing it through the atomizer. Atomization will give the drug into droplet forms, and in the next slide, as you can see, so the drugs will be the itraconazole plus polymer. Polymer, maybe there are many polymers. For the pharmacists and the formulation scientists, polymers are things that if we do not know about the properties of the polymers, we are nowhere. So there are many polymers: water soluble polymers, water insoluble polymers, pH dependent polymers. There are so many parameters are over there. Now we have to choose the right polymers. Hypromellose is the commercial name of hydroxypropyl methyl cellulose. Its commercial name is hypromellose. And when you see a part phthalate, whenever you say phthalate, say cellulose acetate phthalate, cap, say shellac, say Jane, all those are designed as a pH dependent polymer. Diclofenac sodium and NSAID group drugs, which actually create a lot of damages to the stomach. I can remember one thing. When I was before joining Dhaka University as a lecturer in 1992, I thought that why not go to some pharmaceutical companies and get some experiences from there that will actually help our students. So I joined the Sanofi Aventis. That was that time in 1990. It was called Ron Polenk Rohrer. And one day I was working in the production section, and a senior brother was working in quality. Then we heard that he actually vomited blood, actually, because he was taking diclofenac sodium for months, not with food and not even with any PPI or supporting. So the thing happens, you know, diclofenac sodium releases only 10% of diclofenac sodium, because it is an acidic drug in the stomach. It remains mostly non-ionized, so only 10% drug is released, and that 10% was enough to create this perforation on the lining, and result he was vomiting. And luckily from Tongi we took him to Holy Family Hospital back in 1991. I can't remember the exact date. And now he is in France and doing excellent. Okay, so I have given you the idea that this technology is one of the technology. I tell you, this spray dyeing is one of the technologies. There are other technologies also. Then this is the thing that I already discussed. Crystalline structure, more rigid and compact due to multiple bonds. In crystalline structure, there are many bonds, so before it gets dissolved, we have to break down those bonds, right? So as we can understand, that will be very strong, and that's why solubility is less. And when we are converting itraconazole with polymer by technologies like spray drying, we are getting the amorphous structure. This is less rigid and compact with covalent bond only. Covalent means equal sharing of electrons. This is very feeble, means it's very weak bonds. We can easily break it down. But the ionic bonds and other bonds, those are very difficult to break. So that's why amorphous structures are easily highly soluble and more effective. These are some parameters regarding the area under curve, and we can see that some of the companies like Tolsura, they are actually adopting this 65 milligram SUBA technology, and twice is 130 milligram. So their area under curve is compared. So in brief, in general, to make the sense is that conventional 100 milligram, after so many things giving rise to the AUC curve, the SUBA technology, we are taking only 65 milligram, and we can overcome all the difficulties. Means the journey of the itraconazole we have to understand. After swallowing, it goes to the stomach, and from the stomach it descends down to the small intestine, and through the small intestine by transcellular or paracellular and other pinocytosis, exocytosis. There are so many mechanisms, and by those they get available, right? Now, what are the benefits of this SUBA technology in general? 90% bioavailability, maximum efficacy in a smaller dose, 65 milligram, and no interactions with PPIs. Means in the stomach, when co-administered with PPIs, then if it is not protected, then PPIs will probably render a little bit slow those conventional dosage forms. I will talk because you have seen that we are using hypromellose phthalate. I told you already. Hypromellose means hydroxypropyl methyl cellulose. This is a hydrophilic polymer, and it is the best one. Why? Because if you prepare with hypromellose, even a sustained release dosage form, it gives rise to a zero order release, which is actually our dream, that a drug will release with zero order, concentration independent. When the tablet was full of the itraconazole, it will be released that way, and at last also released in the same way, if we go for that. So these types of polymers, they are giving the advantages, and unabsorbed drug accumulation is very less. Let me clarify one thing. What is the advantages of using this phthalate? As the pH of the stomach is 1 to 3, and when we are using hypromellose phthalate, the matrix structure will be coated by this phthalate, right? And this is pH dependent, means at this low pH nothing will happen to phthalate. So the drug along with the matrix will come to the intestine as it is. There will be not much drug inside the stomach. So we are not going for losing the drug inside the stomach. Rather, successfully we can, after emptying the stomach, after the residence time, the drug along with the polymer matrix will come to the intestine and will start functioning as if we have directly given the drug to the small intestine. As you know, the small intestine is the natural hub for absorption. Its surface area is enormous in comparison to the stomach. That's why most of the drugs, whether acidic or basic, they actually get absorbed from the small intestine, right? Conventional itraconazole crystalline structure, this is amorphous. There are some repetitions. Conventional 100 milligram, this is 65 milligram. And then interaction with PPIs 68% less bioavailable, and SUBA no interaction with PPIs, because there is no chance of interaction. We have actually coated the molecule with the phthalate. Phthalate is always pH dependent, and it is not dissolved when the pH is above 6. It is only dissolved. So the drug is get protected. and unabsorbed drug, more accumulation and causes side effects and less accumulation. Thank you very much.